Hello and welcome. My name is Dr. Kishore Managoli, and from the archives of our Legion Library, we'll see how to detect apoptosis with flow cytometry. Now, this is going to be a very short video, and I hope you'll enjoy it. The best way to understand it is by understanding a clinical scenario. So we have a 52-year-old male from Kenya who is diagnosed with yellow fever. Liver biopsy shows many Councilman bodies. If you recall, Councilman bodies represent apoptotic bodies. A flow cytometry analysis using annexin V and propydium iodide is shown in the scattergram here. Now they are asking two questions. One is that what do annexin V and propydium iodide detect? And the second part of the question is how do they work? Now before we move to that part, let me explain this scattergram to you. So we have the x-axis represented by annexin V. The y-axis is represented by propydium iodide. The scattergram itself consists of 4 quadrants, as you can see. The lower left quadrant, the upper left quadrant, the lower right quadrant, and the upper right quadrant. So how do we detect apoptosis and how do we interpret this scattergram is what we are going to look at. So the left lower quadrant represents live cells. The left upper shows necrosis. The right lower shows early apoptosis, and that is what is detected by annexin V and propydium iodide. And the right upper is represented by late apoptosis. Now, this answers the first part of your question as to what they detect. They detect early apoptosis. So this is a very important point for you to remember. They detect early apoptosis and not late apoptosis. So this is something worth remembering. Now, the second part of the question is how do they work? So this is an image which explains the principle of the annexin V assay. So we know that the cell membrane, the outer cell membrane of a cell is made up of a phospholipid bilayer. And the outer surface, the external side of the cell membrane, is composed of phosphatidylcholine and sphingomyelin, whereas the internal side of the cell membrane also contains what is called as phosphatidylserine, which is not present normally on the external side of the cell membrane. However, the story reverses in apoptosis, and this is the most interesting part of this process, in that phosphatidylserine suddenly flips to the external surface of the cell membrane. And that is what makes it very interesting. Now, phosphatidylserine up till now was hidden on the inner side, and it was invisible to especially the macrophages that are roaming around in the cytoplasm of the cell. Now, the moment they sense or they smell a phosphatidylserine, macrophages will come and gobble them up because they find phosphatidylserine to be very yummy. And the moment they gobble up these phosphatidylserine moieties, it leads to disintegration of the cell membrane and apoptosis. And also, interestingly, this annexin V assay binds to exactly this phosphatidylserine, so it becomes easy to detect early apoptosis with the help of this assay. So it's important for you to understand the principle of the annexin V assay. Similarly, for the propydium iodide, it has a different mechanism where you have a live cell. We saw that annexin V utilizes the outer cell membrane. Now, propydium iodide uses exactly the opposite thing. It dives deep into the cell, right into the nucleus, and binds the DNA of the dead cell. And this is how we can detect both the death of the cell through DNA binding as well as death of the cell by binding to phosphatidylserine of the cell membrane. So this is the summary slide which you can use for a quick recap. Thanks for watching.